wanting to talk to you about cell-mediated immunity. We left off looking at the two types of specific immunity, cell-mediated and antibody-mediated. So let's talk about cell-mediated immunity and the three main steps. Step 1 is antigen presentation. Step 2 is antigen recognition. So it's like step 1 is when you're faced for the first time with the antigen. It's being presented to the immune system. Step 2 is recognizing the antigen. Step 3, part A, is activation of the CD8 T cells. And then step 3, part B, is activation of the CD4 T cells. Okay, so let's look at step 1, or antigen presentation. The antigen must be bound to glycoproteins. Glyco means carbohydrate, and proteins means protein. So this is a molecule that's a combination of a carbohydrate and a protein in the cell membrane. They have to be bound to that in order to activate the T cells. These glycoproteins are termed MHC proteins, which stands for major histocompatibility complex proteins, or human leukocyte antigen proteins. So we just abbreviate them and say MHC proteins. They're sometimes called HLA proteins, but we'll call them MHC proteins. There are two main classes of MHC proteins. Okay, we're still in step 1. We've got class 1 and class 2 MHC proteins. All right. So before we go any further, let's look at and go back here and talk about class 1 MHC proteins. These are found on cell membranes of I'm going to just type this kind of small. They're found on cell membranes of all nucleated cells, meaning it would not be found on red blood cells because red blood cells are not nucleated. They're made in the endoplasmic reticulum, and they're transported to the Golgi apparatus, and then what they'll do is they will pick up small peptides, they'll transport them to the cell membrane, and they'll insert these little peptides into the cell membrane attached to these class 1 MHC proteins. These little peptides are ignored by the body if they are normal, but if they are non-self proteins, they will cause T cells to be activated. Class 2 MHC proteins are found on only the surfaces of antigen-presenting cells, or APCs. And I should make these a little bigger, which I will just do for you right quick. And you can read your lecture notes to get a little bit more information about these class 1 MHC. And please, I do recommend you read your textbook on these as well. But you've got to remember that the class 1 MHCs are found on all nucleated cells. The class 2 MHC proteins are different. These are found only on the surface of antigen-presenting cells, or APCs, which would include macrophages and dendritic cells. Dendritic cells are cells that are found in certain tissues like the spleen, etc. So again, certain lymphocytes will also have, it will also be can be antigen presenting cells. But basically, only, or excuse me, I take that back. What I'm saying is that class 2 MHC proteins are found on macrophages, dendritic cells, and some lymphocytes. Okay, so let's kind of move here and look at what happens when a virus or a bacterium invades the body. Well, there are two separate events. First of all, a cell is infected. The cell is infected by the organism. So this will stimulate the killer T cells. These are the CD8 cells. These are T cells that are also called CD8s, and I like to call them the haters because 8 sounds like hate. These are the killer Ts, but let's more officially call them the CD8s. Now, at the same time that the cell is infected by this foreign organism, then a macrophage or antigen-presenting cell will encounter and phagocytize another one of these exact invading pathogens. They will see this organism, this virus or this bacterium. They'll eat it or phagocytize it, and then they'll say, "Hey, look!" And I have this written out for you. Look at the bad guy I found. Let's go get help and get rid of him, so that this will help activate the helper T cells. The helper T cells are the CD4 cells, and I like to think of them as help for you. So we have two separate things happening. We have a cell that gets infected, which will stimulate the CD8s, and meanwhile, at the same kind of general area, a macrophage will see the same invader organism, and it will phagocytize it, and it will then cause the activation of the CD4 cells. These are the helper T cells. So here's a picture of our infected cell. There's the virus. There's its little protein coat. It's injected its genetic information here. This is a bacterium. So we're showing you a virus and a bacterium. And these little abnormal peptides, which are the antigens, are going to be then taken in. They're going to be taken up by the class 1 MHC protein. It'll incorporate this little abnormal peptide and hold it out there. And then, of course, in the Golgi apparatus, it's going to be put into a little vesicle. The vesicle will carry it to the plasma membrane, and it will then put this little protein out on the surface and say, look at me, I've been infected, and here is the abnormal peptide. Okay, this is an infected cell. So the question is, what actually holds the antigen on the cell membrane of an infected cell? Is it the phospholipids of the plasma membrane? Is it class 1 MHC protein? Is it class 2 MHC protein? Is it the CD8 marker or the CD4 marker? And I'm going to let you think about that yourself. I think you know the answer. Okay, and this is what's happening also with this infection. And here we're showing the bacterium that's been phagocytized by the antigen-presenting cell, which would be like a macrophage. It's taken in and by endocytosis. It's then digested by a lysosome, breaking it down, producing fragments. The antigenic fragments are then bound to a class 2 MHC protein, a different MHC than the previous. And the antigenic fragments are then carried, again, the whole process, just as we've seen before, where this particular protein, or the MHC2 protein, will take a hold of the antigen, and then it will travel and go onto the plasma membrane, and it will be placed out there saying, look what happened, look what I did, I engulfed this invader, and here's the protein, and we got to tell everybody, we got to tell the world that we've been infected, so we need to do our job here. So that's what the antigen presenting cell does. So step 2 is antigen recognition. Antigen recognition. So this is where CD markers come in. The CD8 markers are found on the cytotoxic or killer T cells. Cyto means cell, toxic means toxic. These are the T cells that are also called CD8 cells. So the CD8 markers are found on CD8 T cells. Okay. Suppressor T cells are also called CD8 T cells. So we've got these. These are the haters, right? These the cytotoxic and the suppressor T cells. Okay. These cells, the CD8 T cells, will respond to antigens that are presented by class 1 MHC proteins. So the CD8s respond to antigens that are presented by class 1 MHC proteins. And the CD4 markers are found on helper T cells. These are called the CD4 T cells. And the CD4 T cells respond to antigens that are presented by class 2 MHC proteins. So let's look a little bit. Here's another question. The CD8 cells are going to respond and bind to antigens that are presented by MHC1 proteins or on infected cells, antigens that are presented by MHC2 proteins on an APC, antigens that are free in the cytoplasm, or antigens that are located in the nucleus. I'll let you answer that one. Okay, we have now the process called co-stimulation. Co-stimulation is a process that is a secondary binding process that is required to confirm an initial activation signal that's started by antigen recognition. It acts like the safety on a gun. It prevents accidental activation. So co-stimulation proteins like cytokines that are produced by lymphocytes are required to activate T cells. So co-stimulation. So we have pictures of co-stimulation that is shown right here. Right here is an infected cell. The infected cell has got its MHC class 1 protein in the membrane. He's got his little antigen. Here comes the CD8 cell. He's holding out. He will bind to this antigen here because he's a specific for this antigen. But before this will happen, I don't know why it does that. Before this will be activated, this infected cell will have to secrete some special chemicals, lymphokines or cytokines, to cause this co-stimulation to make sure that this should be happening. Okay, so here's step 3A, the activation of the CD8 T cells. The cytotoxic T cells, or the killer T cells, these guys destroy the abnormal and infected cell. They will kill the cell. So the cell that's been infected is a dead cell. He gets killed so that he can't, so that that infected cell cannot keep producing viruses or keep cranking out more infection. The memory T cells are actually the memory cytotoxic T cells will remain inactive. These are a few cells that will remain until the same antigen might appear the second time. And then we have suppressor T cells. They're also they're called T sub S cells, and these will secrete suppression factors or cytokines that will slow down the immune response and help keep it under control. All right, so that's step 3A was the activation of the CD8 T cells. And this is a picture showing you this process. There's the inactive CD8. Here's the infected cell. These are the little antigens. The antigens are being held out by the class 1 MHC protein. And this CD8 cell has a T cell receptor and that is specifically shaped to bind only with that antigen. And when this will bind together, then we're going to have the co-stimulation occurring, and then we will get this guy activated. Once it's activated, the T cells are activated, they're going to divide and make lots more of them. And then a few of these are going to remain in the tissues in order to be memory cells that will be inactive just in case there's another infection of the same antigen in the future. Okay, this is what happens when this is the infected cell, this is the whatchamajigger, sorry, this is the cytotoxic T cell, and we're kind of showing it from a slightly different angle. This is the killer T cell. This killer T is going to release lymphotoxins, which will disrupt the cell metabolism and kill it. It will release cytokines, which will stimulate apoptosis, which is cell death. And it can release perforins, which will cause perforation of the cell membrane or destruction, which will then allow water to come in and it will break down and lyse. And then the target cell is destroyed. And there's our killer T cell, our CD8. Step 3B is now the activation of the CD4 T cells. The CD4 T cells. Now, the activated CD4 T cells produce what we call helper T cells. Help for you, I like to think of. The CD4s are help for you. And they will also produce memory T cells that will stay in reserve for the next exposure to the antigen. Here's a picture. There's the antigen presenting cell that has taken in the foreign antigen. There it is right here. This is the inactive CD4 or helper T cell. We've got a blow up of this little interaction here. This is the class 2 MHC protein that's holding out the antigen. And then here is the, so it's right here holding out the antigen. Here's the inactive T cell, this guy right here. This is him blown up. And it's showing that he's got the T cell receptor, which is shaped specifically to bind only with that antigen. And then here's the CD4 protein. This is the marker. This is the CD4 marker protein that designates this as a CD4 T cell. All right, once this activation occurs, then these CD4 cells will start to release cytokines. These cytokines are going to do lots of things, and these are going to do cell division. We're going to get lots of these activated helper Ts. We're going to have some of them remain as memory helper Ts that are inactive for a future infection. So let's look then at this cell-mediated immunity, this summary. The virus-infected cell will show his antigen that has been taken in on a class 1 MHC protein. This will activate the CD8 cells, the cytotoxic or the killer Ts, to destroy the infected cell. The antigen-presenting cells, meanwhile, will take in the antigen and then they will display it or show it on class 2 MHC proteins on their cell membrane. And this will activate the CD4 cells, the helper Ts, to stimulate the immune response by the T cells and the B cells. Both the cytotoxic Ts and the helper Ts will produce memory cells for the next exposure to that same antigen down the road. So the question is, helper T cells, do they have CD4 markers? Do they respond to antigens presented by MHC class 2 proteins on APCs? Do they secrete cytokines that stimulate T and B cells? Do they divide to form memory helper T cells for future exposure or is it all the above? Okay, this is a picture showing you this process of the activation of the CD8s. So this is the CD8 T cell activation by an infected cell, and it tells us that we get the cytotoxic T cells, we get memory T cells, and we get suppressor T cells. Remember, CD8s will divide to form cytotoxic T cells, or T sub C, memory cytotoxic T cells, and suppressor T cells. All right, the next thing that happens is that these antigen-presenting cells will cause the activation of the CD4 cells. So again, they hold out the antigen on the class 2 MHC protein. We've got the CD4 cells getting activated. They're going to produce helper Ts, which are going to stimulate a response by the T cells and B cells. And then we have the memory helper Ts that are going to wait for the appearance of the antigen again down the road. Remember, CD4s will divide to form helper Ts and memory helper Ts. Okay, so we've finished cell-mediated immunity, and again, you're going to need to look at your textbook carefully, look at your lecture notes, and work through the activities that we do in class together. So, sorry this video was a little long, but I just wanted to keep all of the cell-mediated immunity together. Thanks.